Hey there, I'm going to show you how to put together and fill in your DNA foldable. So basically what we need to do is we need to start with 3 blank pages from our packet. So I have 3 pages here, and I'm going to actually move my notebook out of the way for just a second. And what I'm going to do is just kind of stack the 3 pages on top of each other like this. So I'm creating these little headings. So there's enough space here where I could, write a title if need be. I don't want it to be too big or too small. So there are my 3 pieces of paper, okay? So I've got my 3 papers, so top, middle, this one's on the bottom. So I'm going to take my paper and I'm going to flip it up on top of itself like this. Let me redo it. So here's my 3 pieces of paper, right? Top, middle, here's the one that's on the bottom, okay? And then I just flipped the paper over on top of itself, and now I'm going to fold it over on itself, right, like that. Okay, now I'm going to grab my stapler. So I'm going to grab my stapler, and I'm just going to put just 2 or 3 staples into my foldable. There's the end, there's the middle, and then one again on the other end. So now I've got my foldable right like this, and this is perfect. I've got 1, 2, 3, 4, 5, 6 flaps. So what I'm going to do for my DNA foldable on the front cover right here, say DNA stands for deoxyribonucleic acid. So there is my cover, and I wrote this in a different color so that when I'm flipping through my notes, it kind of stands out. And then I'm going to go ahead and I'm going to title each one of these flaps. So DNA is made of, and the next one is location and job, and then structure, replication, and then finally a drawing of DNA. So I now have my titles for each of these. So let's start with the made of section right here. So I'm just going to flip this up and kind of fold it over just for a second. So I'm starting with the made of right here, and we know that the monomer, or the building block of DNA, is our nucleotide. So I'm going to grab some glue, and I'm just going to glue down my nucleotide. All right, this is from right here, goes on with our packet. All right, so my nucleotide, right, is made up of a phosphate, and normally we would say just sugar, but specifically it is a deoxyribose sugar because this is DNA, and then of course a nitrogen base. And then DNA is a nucleic acid, right? We're just taking some notes. So DNA is a nucleic acid made of repeating monomers called nucleotides. And then there are 4 types of the nitrogen bases, which we need to be familiar with. We have adenine, guanine, thymine, and cytosine. So these are the notes for this flap, for what is DNA made of. So I'm going to go to the next flap where we're talking about location and job. So I'm just going to flip this up, kind of crease it down. There we go. All right. So taking a few notes on this page. So DNA is a recipe book that contains the production or synthesis of proteins. You may have heard of DNA as a blueprint to life. That's not actually accurate. It is more like a recipe book where you go in a recipe book and you get ingredients, eggs, milk, sugar, right, to make a cake. For our body, right, we go to the DNA for the recipe or the ingredients to make proteins. So DNA is our recipe book. And then as far as its job, its location, that's the job, is to be the recipe book. The location, DNA is located in the nucleus of eukaryotic cells. All right, you and I have eukaryotic cells, therefore our DNA is located in the nucleus. Prokaryotes, it would be located in the cytoplasm. All right, so that's it for this flap. So we're going to move on to the next flap, talking about structure. I'm going to flip up my flap right here. All right, I'm going to need another picture. I'm going to need this picture right here that I've cut out. So I'm going to grab my glue again. Go. I'm just going to glue this down into my foldable. There we go. Beautiful. Look at that. All right, so I got my DNA. So the structure of our DNA, we're going to label a few things here. So first, we've got our 5-carbon sugar, right? We know it's a deoxyribose sugar. The next thing we're going to label is our nitrogen base, right? That can be either A, T, C, or G, adenine, thymine, guanine, or cytosine. Now, the nitrogen bases are held together by a hydrogen bond. It's a pretty weak bond. And then the third part of our nucleotide is the phosphate. Right, so that's the 3 parts of our nucleotide: phosphate, sugar, base, and then they're all connected together to give us the entire nucleic acid. So as far as the actual structure, DNA is made of 2 strands of nucleotides. DNA's shape is called a double helix, sometimes a twisted helix. And then DNA will always follow a base pair rule when it comes to how the bases will line up with each other. A will always pair with T, and G will always pair with C. So adenine with thymine, guanine pairs with cytosine. And again, the bases are held together with a hydrogen bond. Look, I said it twice. It's probably important. Who knows? So the structure, again, we've got these repeating nucleotides, right? So if I put a box, right, here's one nucleotide, phosphate sugar base, and then I could put another box, you know, right above that around the phosphate sugar and the base. So my structure, right, it's two strands of our nucleotides, the left side and the right side. All right, that's all we have for our structure of our DNA notes. So let's flip to the next page, talk about DNA replication. I need my last picture here that I cut out, one that looks like this. So I'm going to grab that picture and glue it down to this next to last flap. There we go. Get some glue. And there we go. All right. So let's talk about DNA replication. So DNA has to replicate before a cell divides, right? Before a cell divides. The goal of DNA replication is to make two identical strands of DNA. Right? We want them to be identical. We don't want any mutations or any new strands of DNA. They need to be identical. So there are a couple of steps to DNA replication. Steps of DNA replication. All right, so number one, sorry, my writing surface is a little iffy. All right, so step one, the double helix, right, the structure, the double helix is going to unzip or unwind. And when that happens, the hydrogen bonds are broken. So we are unzipping the double helix, breaking those hydrogen bonds. The complementary nitrogen bases in the nucleus will start to bond to the unzipped strands. So we're breaking apart that bond between adenine and thymine. So that free adenine that's now, hanging here off of the DNA, a thymine will come and it'll attach to it. Once it's finished, two new DNA molecules are produced. And so overall, DNA replication is semi-conservative because each DNA molecule produced has one strand from the old molecule and one new strand. So what we mean by semi-conservative, if I were to take this picture and kind of add some color to it, so I'm just going to kind of redraw it right here. Yeah, there we go. Okay, it's the best I can do. So I've got my original strand, right, or the parent strand. So that's the original strand. And then, right, so I've unzipped it. So here it's zipped together, here it's unzipped. So complementary nitrogen bases are going to start attaching themselves to those unzipped strands to create two new strands of DNA. And so now this blue strand, right, that is the new strand. So it's semi-conservative because eventually these will not be attached at all, right? It'll be two completely separate DNA strands because eventually we'll reach the end. And so each new DNA strand is made up of one strand from the original molecule and then one new strand. So that's why we say DNA replication is semi-conservative. And then finally, here at the end on our last page, we need to include a drawing of our DNA molecule. So I can just kind of take my pen and I'm going to make my double helix or my twisted ladder. Like that. And then I need to add in, all right, my bases, nucleotides. All right, there we go. Look at that. That is the best DNA molecule I have ever seen. Yours is probably going to be a little bit better than mine. That is it for our DNA foldable. If you still have any questions about this foldable or the job of DNA, the structure, replication of DNA, please be sure to let me know.